I'll introduce our next speaker, which is Professor Vatsan Raman from the University of Wisconsin-Madison. Um, and he previously worked at the Weiss Institute in Dr. George Strich's laboratory, working on engineering microbes for biosensors and sustainably producing fuels and chemicals. And now does some super interesting work on phages and engineering them and their interactions with the microbiome. So you can go ahead and give your talk then.
All right, can you guys see the slides okay?
Yes.
That's good. And can you see the gigantic pointer?
Yes.
All right. All right. Super. Well, thank you for, and you can hear me okay too? No problems? Okay. Well, thank you for the invitation. It's such a pleasure to be here. So, as was just mentioned a couple of minutes back, my name is Vatsan Raman. I'm an assistant professor in the University of Wisconsin-Madison. And my lab works on a couple of different things. But what I'll do today is present our work on understanding and engineering bacteriophages and their use as potential antimicrobial therapeutics. So, before I get started, okay, before I get started, I want to acknowledge all the people in my lab. It is such an honor and a privilege to be their mentor and to be here and present their work to you. Okay, as biochemists and as synthetic biologists, our overall goal really is to understand the molecular rules of life. And to achieve this goal, we pose broadly speaking, two types of questions. The first type of question is what I'd like to think is one of mechanism. And the mechanism question really seeks to explain how different biomolecules interact with each other to carry out some cellular process. The second type of question that we pose is one of function. And what the function question does is it seeks to explain what is the molecular basis of function. In other words, if you think about a protein, it seeks to explain what is the role of every residue of a protein towards protein function. And what I would argue is over time, we've become really good at understanding the mechanism of different cellular processes. In other words, we've become good at wielding a scalpel, so to speak, to sort of deftly peel off layers of different biological processes and understand the mechanism. And so you can pick any cellular process, transcription, translation, signaling, transport. We understand these at great depths because the classical reductionist approach to understanding biology works really well. Now, we've also made significant advances in understanding the molecular basis of function, and structural biology tools such as cryo-EM and NMR and crystallography play a major part here. And yet I would argue there's a lot that remains unknown. And the reason for this is, structural biology tools give you, well, a structure, right? And going from structure to the molecular basis of function, that's not really a straightforward path. But let's pause here for a second and ask, well, what do I mean by molecular basis of function, right? If you take any arbitrary protein, right, the overall function of that protein, this could be an enzyme, this could be some binding protein, the overall function of the protein is really the collective output of every residue of the protein, right? And so when I say molecular basis of function, what I simply mean is what is the contribution of every residue of the protein towards its activity, towards its specificity, affinity, stability, allostery, etc. And if we want to answer the question, what's the molecular basis of protein function, we have to understand how sequence variations impact function. In other words, how systematic changes to a protein sequence impacts function. But this question, how do systematic changes to protein sequences impacts its function, it really gets to the root of a problem in protein science or biomolecular science that we often tend to ignore or at least forget. And that is the quote unquote bigness of protein sequence space. Now, what do I mean by that? If you look back and ask, what have we learned from decades of genetics and biochemistry? One common theme is that even a single mutation to a protein or two mutations to a protein can have profound impact on function, right? And you might have seen this with enzyme assays that you may have done. But let's stop there for a moment and examine what is the actual sequence space of these variations, right? So let's imagine that you had some arbitrary protein which is 100 amino acids long, right? And then you said, well, what do I want to change every residue of the protein to all remaining 19 non-wild type positions and measure the function, right? And so what is the sequence space of these changes? That is 19 times 100, which is 1900 different mutants. And so if you're doing traditional biochemical assays or genetic assays, this is a lot of mutants to measure, right? Or functionally measure. Now you can take it a step further and say, well, now I want to do double site saturation mutagenesis, which means that I want to introduce at every pair of residues in the protein, I want to introduce all remaining 19 amino acids and see how it impacts function. Well, the sequence space of these changes is upwards of 2 million mutations, right? Which is a staggeringly large number. And again, you can logically take it a step further and say triple site saturation mutagenesis, and that's upwards of 1 billion sequence combinations, right? And so you see the sequence space of protein variations that explodes very quickly. And you may have already, but if you step back and see, well, how does nature do its business, right? But nature samples all mutations at all positions all the time. And you've probably seen evidence of this when you blast search your favorite protein, right? So when you blast search your favorite protein, you might find that there are hundreds to thousands of sequence homologs that match your protein. And these sequence homologs might be anywhere from 90% sequence identity to 20% sequence identity, right? Which then begs the question, well, how are they functionally different from each other, right? Do these sequence variations, does it encode new activities for the protein, new specificities, different stabilities, different allosterities? And so we really have no answers to these questions. And so I would argue that if we want to answer the question, what's the molecular basis of function, we have to start making functional measurements at the scale at which nature samples them. And we have to start making functional measurements at the scale at which we're discovering new protein sequences from genomic databases. And so this traditional idea of making one or a few, measuring the function of one or a few mutations at a time to understand function, I mean, that idea is simply not going to cut it, right? And so I want to illustrate this point with a simple example here. So let's imagine that this is some sequence function landscape, right? So on the x-axis, you have some sequence variations of a protein, and on the y-axis, you have some measure of function. And so this is some sequence function landscape. When we pick out, one or a few candidates to functionally characterize, what we really see is a small sliver of the sequence function landscape, right? And the example that I like to give is this old parable of 5 blind men and the elephant, and some of you may have heard of this, right? So there are these 5 blind men who touch different parts of the elephant, and they want to make sense of what the elephant looks like, right? But they touch these local regions, and they have no idea what the elephant looks and feels like. And likewise, as you sample one or a few sequences, you see these tiny local regions of the sequence function landscape, but you have no idea what it looks like in totality. But let's imagine that instead of 5 blind men, you had 50 blind men, right? Now, collectively, these 50 blind men have a slightly better sense of what the elephant looks like, right? And likewise, as we start functionally sampling more and more sequence variations, you start seeing the peaks and the valleys of the sequence function landscape. Now, let's imagine instead of 50 blind men, you had 500 blind, right? Now, collectively, they have a complete sense of what the elephant looks like. And likewise, as you start functionally sampling more and more sequence variations, you, you get the full picture of the sequence function landscape. And what that then means is that we can then figure out what the fundamental molecular rules are that govern protein function, which we could then use to engineer new functions into proteins for synthetic biology applications. So which then brings me to what my lab does. So the overall goal of my lab is to generate and understand these sequence function landscapes, and to then use that information to figure out the basic rules that govern protein function, and to then design proteins that have novel function. And so to do this, what we do is we introduce mutations in proteins in very high throughput, and we make functional measurements to proteins in very high throughput. And so the question is, well, how do we make functional measurements in high throughput? And so the way we do this is to use next generation DNA sequencing, also known as deep sequencing, to make functional measurements. I'm going to do a small detour for a couple of minutes, and then we'll get back on track. So in the field of computer processors, there's something called as Moore's law. And so this was named after Gordon Moore, who was the former CEO of Intel. And what Moore's law states is that the power of computing doubles roughly every 2 years, or the cost of computing halves roughly every 2 years, right? And so this is a Moore's law plot. Now, if you think about that for a second, right, that is explosive technological growth. And there's besides computer processors, there's really no other technology that matches the growth of Moore's law, except next generation DNA sequencing. And so this is a plot of next generation DNA sequencing. And next generation DNA sequencing not only beats Moore's law, it beats Moore's law by a mile. And so this is roughly 2005 when next generation DNA sequencing was invented. And this resulted in a precipitous drop in the cost of sequencing, and which ushered in what I like to think of as a sequencing-driven revolution in biology. Now, what does this have to do with protein sequence function landscapes? I'm going to get to that in a second now. And so the sequencing-driven revolution in biology really unfolded in 3 stages, if you will. The first stage, which is, again, starting roughly in 2005, is I like to think of this as the compositional measurements, right? And so here the idea was we use deep sequencing to measure the genomes of different animals to figure out molecular phylogenies. We sequenced human populations to understand the genetic basis of diseases and other traits. We sequenced microbiomes to figure out the composition of bugs that live within us. So this is the compositional phase. The second phase is what I like to think is the observational phase. And so there's an entire alphabet soup of different seqs, like, RNA-seq, ChIP-seq, methyl-seq, ribo-seq. And so these technologies use DNA sequencing to measure different cellular processes, chromatin state, transcription factor binding sites, genome structure, etc. The third stage, which is starting roughly in 2015, is what I like to think of as the perturbational phase. And here the idea was to introduce mutations in high throughput and use sequencing to make functional measurements in high throughput. And so this is where I'd say where my lab makes a contribution. It was roughly around this time when the CRISPR-Cas9 and other gene editing tools were coming out. And what we figured then was, if we can combine the ability to introduce mutations in high throughput and make functional measurements in high throughput, well, in principle, we can reconstruct these sequence functional landscapes. And so that's what we did. It turns out that DNA sequencing is an ideal readout for functional measurements, because the volume of sequences, volume of sequencing scales with the quote unquote bigness of protein sequences, right? And so a typical sequencing run could produce hundreds of millions of sequencing reads to a few billion reads. And so in principle, if you can couple biomolecular function to sequencing, well, then you can systematically and comprehensively map the sequence function landscape. And so to do this, we take a few different approaches in my lab. We can connect the protein function to a reporter gene. And so the idea would be different mutants would activate the reporter to different degrees. And so we can sort the reporters into different fluorescent bins and sequence the bins. And then that's how we can connect function to activity. Or we can use a barcoded transcript. And so we can do cDNA synthesis and then sequence the cDNA. So that is a group of measurements called as the reporter-based measurements. Another option would be to link the protein mutants to host fitness or cell fitness. So some mutants would benefit cell fitness, some mutants would hurt cell fitness. And so you can quantify the population before and after, and then you have a score for each protein mutant. And the third approach that we're beginning to investigate is how we can use single-cell RNA sequencing as a tool to ask how different mutants impact the transcriptomic state of the cell. Now, we take these general principles and we apply them to study three different problems in my lab. One is to understand and engineer allostatic proteins. And so these are proteins which, when there is a binding event that happens at one end, there is an activation that happens at a distal end. And so we can use these as biosensors in synbio applications and other such uses. We use them to study bacteriophage host interactions and to engineer synthetic bacteriophages as potential therapeutics. And the third problem that we look at is what is the functional impact of disease-relevant or mutations in disease-relevant genes found in human populations. But for today's presentation, what I'm going to do is I'm going to focus only on the bacteriophage host interaction aspect of my lab's work. Bacteriophages are pretty remarkable creatures, and I'll mention one number, which is just a staggeringly large number. They're the most abundant biological entity on Earth, and the estimates are that there are 10 to the 31 phages on Earth. So just so think about that number for a second, right? 10 to the 31 number. And you can imagine these to be the longest evolutionary experiment that nature has conducted, right? Because there's this constant arms race between bacteriophages, which are microbial viruses, and their hosts, right? And so there's this back and forth, and you can imagine that to be the longest evolutionary experiment. Now, bacteriophages have very high sequence diversity and high genome mosaicity. And this allows them to adapt to different environmental niches and different host conditions very quickly. And it also creates this nice and strong link between sequence and fitness, which we can take advantage of for large-scale functional measurements. Now, I'll mention the life cycle of a simple lytic bacteriophage, and it really involves sort of 4 steps. And so this is the bacteriophage or the microbial virus. What it does is the first step is it absorbs on the host. The second step is it delivers its genetic material to the host. The third step is, so that's the infection part. The third step is replication, where it replicates, it hacks the host machinery and replicates inside the host, makes many copies of itself and disables the host machinery. And finally, it lyses the host and then swims off to infect other hosts, right? And so this is the life cycle of a simple lytic phage. And what really sort of got us interested in the bacteriophage question is the societal grand challenge that we face in antibiotic resistance, right? And most of you are probably familiar with this, but I'll mention a few charts that caught my attention. So this is a study that was done by the UK government a few years back, which predicted that if the antibiotic resistance problem is not addressed soon, the number of deaths from antibiotic-resistant bacterial infections is likely to surpass cancer in the next 2 to 3 decades. And what I found particularly alarming are these 2 charts. And so this chart here tells you when an antibiotic molecule was introduced in the clinic over the last 6 to 7 decades, and when the first resistant bug emerged in the clinic, right? And what you'll see is, in the 50s and the 60s, a new molecule typically lasted about a decade before a drug-resistant strain emerged. What you'll see more recently is that as soon as you introduce a new molecule, there is spontaneous resistance to the new molecule. And that's because these bugs already carry resistance gene cassettes that they've taken up from the environment. Now, the other problem is that the number of drug molecules that are coming through the FDA pipeline is dwindling, right? And so we have this double whammy situation where on the one hand, we're not discovering new molecules. And on the other hand, those that do make it through don't last too long in the clinic. And so we said, well, how about we come up with a completely brand new idea of treating the antibiotic resistance problem? And that is to use bacteriophages, right? Now I'm sort of I'm kidding there because people have known about the antibacterial resistance property of phages for almost a century now. And so here is a really neat ad from the 1930s from this place called Le Laboratoire du Bacteriophage, and so this is an ad where they were trying to sell bacteriophages as for treatments against E. coli infection, staph infection, intestinal diarrhea, etc. And so this place, Le Laboratoire du Bacteriophage, eventually became L'Oreal, which is one of the largest cosmetic manufacturers in the world now. So this is the history of L'Oreal. Now, in the West, we discovered penicillin in the 40s, and we just never looked back, right? Because it turned out to be a miracle drug until now. And so I think of phage as the ultimate smart drug for a few different reasons. One is it's highly precise, which means that you could use the phage to specifically remove the pathogen while leaving the rest of the microbial community intact, right? And so in contrast, traditional antibiotics completely wipe the place clean. Second is phages are evolvable, right? Because bacteria will become resistant to a phage as it does to a traditional antibiotic. But the advantage with phages is that in principle, one could use synthetic biology tools to re-evolve the phage to remain efficacious. But unfortunately, with the traditional antibiotic, it's a dead end. The third is it's auto-dose, right? Which means that the phage amplifies at the site of the pathogen. And the fourth is abundance. And what that means is there's just practically a limitless supply of phages against every bacteria, right? So we're never going to run out of candidates to pick from. Now, in the last couple of years, there have been a number of examples where phage therapy has been administered in the United States. And so this is one example that caught sort of public imagination. So Dr. Tom Patterson and his wife, Dr. Stephanie Strathy, were vacationing in Egypt. This was 3-4 years back. And Dr. Patterson ended up getting an infection of Acinetobacter baumannii, a multi-drug resistant strain, and he did not respond to the normal course of antibiotic treatment. So they had to be flown back to San Diego, where they're both professors at UCSD. And so he was then administered a cocktail of phages on compassionate grounds. And this cocktail was put together by Texas A&M University, Naval Research Labs, and a company called Intralytics. And he made a quick recovery. And then after a week or so, there was a resistant infection bacteria that came through, and then he fell sick again. And so they went back into the lab. They discovered new phages, and then they re-administered new phages, and then the infection got completely cleared, and he's been living a hale and healthy life since then. And so there have been a number of other examples, which really then begs the question: well, if phage therapy is as awesome as I make it sound, well, why then hasn't it been widely adopted yet? There are a few fundamental challenges with the use of natural phages for therapy. First is the narrow host range, right? And so I mentioned this as an advantage a few slides back, but it also turns out to be a serious disadvantage, because you can imagine that if there are different strain variations of a pathogen, a phage that might infect one may not infect another, right? And so we have problems with scalability now. The second is the efficacy of traditional antibiotics is much higher than phages, right? And there is a good reason for this, right? Because if a phage is very good at infecting and killing its host, well, the phage itself will perish, right? And so natural selection somehow favors these mediocre phages. And so if we want to achieve high pathogen clearance or to use phages as antibacterial treatments, we have to use synthetic biology tools to remove these evolutionary restrictions to make the phage more efficacious. The third is there is inconsistency in phage cocktails. And so phages, again, are normally delivered as a cocktail of multiple phages. And it turns out that sometimes these phages are highly unrelated, and they have sort of life cycles that sort of don't play together well with each other. And so this results in inconsistency in the performance of the cocktails. And finally, there is bacterial resistance. And as I mentioned before, bacteria do become resistant to a phage. And the only question then is, do we now have methods in place where we can rapidly evolve the phage to remain efficacious? And the answer at this point is no. And so the goal that we set out to solve was to understand how the natural phages work, and to then use that information to then engineer synthetic phages that one could start considering as option for therapeutics. So how much time? All right, so in the next sort of 20 minutes or so, what I'll do is I'll sort of briefly mention 3 vignettes. One is I'll mention a method that we developed for high throughput precision phage genome engineering. Second, I'll tell you how we use that method to do deep mutational scanning of the receptor binding domain of a phage. And then I'll tell you the early steps that we're taking towards making therapeutic phages. And so we decided to focus on understanding the sequence function relationship of the receptor binding domain of the phage, right? And so this is the protein domain that makes primary contact with the host receptor. And hence it governs both the activity, which is how well the phage can absorb on a host, and the specificity, which is what are the different hosts that the phage can infect. And so this is so we picked Bacteriophage T7, which is a very well characterized phage that infects E. coli, and so this is the entire receptor binding domain of the Bacteriophage. And so this little part is the very tip domain that we focused. And so you might recognize this as an IG-like domain. And so an IG-like domain is a protein domain that nature uses over and over again to stick to different things. And so what we wanted to do was we wanted to mutate every residue of the receptor binding domain to all remaining 19 amino acids, right? And so the overall space of mutations is 83 residues, which is the length of the receptor binding domain times 19, which is 1,577 mutants. Now it turns out there are methods for doing untargeted phage engineering, which is using chemicals or radiation, but there are really no methods for doing targeted genome engineering of phages. And part of the reason for that is you have to introduce the gene edits in phages as the phage is actively lysing its host, right? So in the case of T7 will kill the host in 20 minutes, and you have to introduce the mutations into T7 at a targeted location within those 20 minutes. And so this is work done by Phil Huss, who's a talented microbiology PhD student in my lab. And so he developed this method called Oracle, which stands for optimized recombination, accumulation, and library expression. And what it is it's a high throughput, locus specific phage genome engineering method to create large unbiased library of phage variants at a targeted gene. And so it really there are 4 simple steps in Oracle. The first step is to make what we call as an acceptor phage. And so the way the acceptor phage works is we remove the gene of interest on the phage genome. And so in this case, that will be the receptor binding protein. And then we introduce a random piece of DNA flanked by recombinase sites. And so we assemble this in yeast. Now this phage, since it lacks the receptor binding domain, can no longer infect a host. And so here is data that simply shows that once you remove the receptor binding domain, the phage infectivity is down by like 9 logs. And once you recombine in the receptor binding domain, the phage is as active as wild type. So when the bar is small, it means it's highly, highly active. Now, so this phage cannot infect a host. And so the way we make it infect a host is we provide the receptor binding domain in complementation, sort of in trans. And so now we make this chimetic phage, and this chimetic phage now infects a population of cells, each containing a unique gene variant, right? And so this gene variant was synthesized using chip-based oligonucleotides. And it also contains a recombinase. And so now when we activate the recombinase flips in this gene variant at that site that we created early on. And so typically at this stage, we have 1 in 1,000 phages that have recombined. Now, it is very critical that we continue providing the receptor binding domain in trans in order to minimize any library bias that might arise at this stage. Then what we do is we enrich the recombined phages. So I mentioned earlier that 1 in 1,000 phages have recombined. What we now do is we activate CRISPR-Cas9, and then the Cas9 cuts that piece of DNA that we introduced at the very beginning. And so only the unrecombined phages have this piece, and they get chopped up. And so now over time, we can see the accumulation of the recombined phages and the depletion of the unrecombined phages. And so and finally, what we do is then we remove the complemented domain, and the phage then expresses its own mutant variant. And so here are phage variants that express a certain receptor binding domain mutant. And here's just data that shows that our library is well distributed. And so now, the Oracle was really neat because it was locus specific, so we could target any gene. It was highly sequence programmable, so it was not random mutagenesis, so we could introduce any specific sequence, either point mutations or metagenomic sequences. The functional throughput of Oracle was 3 to 4 logs higher than traditional plaque assays or liquid culture assays, and worked on lytic phages. And this is important because the FDA mandates that only lytic phages can be used for therapy. And so now what we wanted to do is we wanted to use this approach to do mutational scanning of the receptor binding domain. And I'll sort of quickly walk you through the idea. So here is the receptor binding domain, residue 1 to residue 83. So we want to mutate the first position to all remaining 19 amino acids. And so that'll be 19 sequence variants. We walk one over. We want to mutate this valine to all remaining 19 amino acids. So that is another 19 sequence variants. And so we do this all the way till the end. And so that's 83 times 19, which is 1577, plus 1 wild type allele, which is present in the mix. And so now what we do is we now use next-gen sequencing to quantify this. And the way we do this is we have an initial population of variants. We apply that on a certain host. Any mutation that is deleterious, the abundance will go down. Any mutation that has no impact, its abundance will not change. Any mutation that is beneficial to the phage, its abundance will increase. And so we can quantify this by deep sequencing. And so I'll just mention one quick number here. Again, out of the 1577 mutants, we found 1573 mutants in the initial library that we constructed. So we had pretty close to 100% coverage of the library. So we applied this on 3 different hosts. And so these 3 are the susceptible hosts. And this is a resistant host. And the resistant host essentially is resistant because it does not have the receptor for the phage. So I'm going to walk you through the data real quick here. So let's look at what the data looks like for application on 10G. So before we look at the whole position, I'm going to sort of walk you through a couple of positions. So here is the amino acid 1 glutamine, and here is the 19 residues. And this is what the data looks like. So each block is a certain mutation of glutamine to say arginine, histidine, lysine, etc. The color of the block is a measure of function. So if it's a white, that means it kind of drops out. That means it's deleterious to the phage. If it is red, that means it is present after selection. The wild type amino acid is normalized to a score of 1, and so that is this. It's marked with this little circle here. And so what you'll see at this position is that it is intolerant to aspartate, glycine, phenylalanine, and tryptophan, right? So those 4 mutations you cannot introduce at position 1. All the other mutations are tolerated to some degree or the other. But what you'll also see is that arginines and lysines have higher fitness than the wild type amino acid. Now let's walk one more position over. So this is position number 2, which is a valine. And here what you'll see that it's really intolerant to most substitutions, right? So everything is white except say alanine and isoleucine, right? And so these substitutions are tolerated. Now we're going to zoom out and look at the full picture. And so here is the PDB numbering of the residues, so 472 to 553. These alphabets here represent the topology of the receptor binding domain. So, B, C, D, etc. And these, B, C, C, D, and E, F are the loops that connect the receptor binding domain. And so now when we look at the entire map, this is what it looks like. And again, I'll walk you through a few interesting features that we found. The first thing is that the receptor bindings, the C-terminal end of the receptor binding domain is a floppy little piece which has no role in structure or function. And so the fact that it was tolerant to most substitutions, it kind of made sense to us because it had no role in structure or function. What you might also see is that the fitness landscape is sparse with peaks, and this is because which tells us that most mutations to the receptor binding domain are deleterious to the phage. What was really neat was that we found phage variants whose fitness was 20 times higher than the wild type phage itself, right? And so this goes back to the point that I made earlier about removing these evolutionary restrictions. And what this shows you is that even a small single amino acid change can remove the evolutionary restrictions on a phage. And you can zoom in and ask, well, what are these changes that are making the phage 20 times fitter than wild type? It turns out that most of these are substitutions to arginines, histidines, and lysines, right? And so one hypothesis is that these substitutions enable the phage. So these are positively charged amino acids, right? So from biochemistry 101. And so these positively charged substitutions enable the phage to interact better with the negatively charged phospholipid group of the E. coli outer membrane. And so that is one possibility for why these might be getting enriched. Now, what you might also notice is that these function-enhancing substitutions are concentrated in specific regions of the receptor binding domain, right? And this is really valuable information from a synthetic biology standpoint, because now if we want to engineer these phages to achieve higher function, we can focus our mutagenesis on regions that give us the most bang for the buck, so to speak. Now, again, this is sort of the overall picture. So I'm just going to take a quick look at the time here. Okay. I'm going to skip a couple of slides. So this is you can zoom in and look at sort of interesting local features of the mutational landscape. So in this case, you see this really interesting sort of alternating patterns of positions that are tolerant to substitutions and intolerant to substitution, tolerant to substitution, intolerant to substitution. And so you could ask, well, what's going on? I mean, can we make sense of why you see this alternating pattern? We can go back and look at the structure of the receptor binding domain and see if we can rationalize why we see that pattern. And so this is the entire receptor binding domain. We zoom into the region that we mutated. And so essentially the regions that are tolerant to substitutions have their C alphas pointing outward, which is into the solution. And the regions that are intolerant to substitutions are positions where the C alpha is pointing inward, which means the side chain of the protein is pointing toward the center of the protein, right? And so you can imagine that any mutation to that region will destabilize the receptor binding domain, and hence it's highly intolerant to substitutions. We can now look at what the data looks like on a different host. This is BW25113. So I'm going to sort of quickly show you the mutational map. So this is the map that we just looked at, which is on E. coli 10G, and this is the mutational map on BW25113. And what sort of jumps at you is that they're not the same, right? So the mutational map on 10G is quite different from the mutational map on BW25113. And so what you might see is that many more substitutions are tolerated on BW25113, but far fewer substitutions are tolerated on 10G. And which is interesting, because what that tells you is that the phage makes unique adaptations even at an amino acid level on different hosts, right? Which again speaks to almost the evolutionary plasticity of the phage, which allows it to sort of change directions as it encounters new hosts in the environment. So I'm going to skip a couple of slides here. Sorry. And so now, so this is the third host, which is BL21. Again, we'll look at the maps, and again, this is a different pattern, which again goes on to show that the phage is able to adapt to different hosts. Now, what we can do is we can take this information and then we can assign a score for every residue of the receptor binding domain as either intolerant to substitution or tolerant to substitution, or specificity guiding, which is these are the positions that are important for specificity on one host versus a different host. And so again, I won't walk through the details here. What I just want to point out is that these regions at the tip are the regions that are the most functionally important regions of the phage, right? So essentially, these are kind of like the fingers of the phage. And so these are the function enhancing substitutions. And so you can imagine these as sort of the tip of the finger, which touches the receptor binding domain, or sort of touches the host receptor. And so if you introduce mutations to these regions, this will likely modify the phage's activity and the specificity of the phage. And again, from a synthetic biology standpoint, that is really useful information. Now, the one question that we wanted to ask was, can we identify gain of function variants in the screen? And what that means is, so a phage normally requires its host receptor to bind. What we wanted to ask was, what if we delete the host receptor? Can we identify new phage mutants that can still infect the host? And so in this case, and so that's what I mean by gain of function variants. So in this case, E. coli, the phages bind to the lipopolysaccharide on E. coli. So we create a deletion mutant which removes the lipopolysaccharide, and the wild type phage has a 7 log loss in activity. And so now when we apply our single mutant library on this host that lacks the receptor, what's really neat is that we find a whole bunch of mutants that can infect the host that lacks the receptor. And so these are what I like to think of the gain of function variants. And what you might again notice is that that these are concentrated around those exact same regions, those loop regions that we looked at earlier, which tells you that that's where the functionally important residue positions lie within that region. Now, and so here's sort of a summary of what the slides, the findings that we had in this plot. What this simply shows you is that most of the mutations that resulted in a gain of function are glycines, which were mutated to histidines, arginines, and lysines at a specific region of the receptor binding domain. And and we clonally test them, and it works really well. And I just want to sort of leave you with sort of a few parting thoughts here. One is that even a humble IG-like domain, so it's not an enzyme, it's not a transcription factor, it's not a transport protein, it's a really simple sticky domain. Even a humble IG domain like has a big story to tell. And what we've already seen is that these phages are able to make sort of really nice host-specific mutational adaptations, and even single amino acid changes to the phage can result in gain of function, right? And what these studies highlight is the power of deep sequencing to resolve these even small functional differences across thousands of mutations. And maybe the last couple of slides, I want to just tell you our early steps at making therapeutic phages. And so we wanted to sort of focus on pathogens, or E. coli pathogens that cause urinary tract infection. So urinary tract infection is a pretty major problem in the United States, and the CDC report of 2019 said that UTI is, community-acquired infection like UTI is a harder problem than hospital-acquired infection. And so one of the problems with phage resistance is that bacteria, when you apply phages on bacteria, there is an initial drop in the bacterial load, but a resistant strain rapidly emerges. And so that actually really hurts the efficacy of phages against bacteria. And so what we wanted to ask was, now that we've done these selection experiments on many different hosts, can we combine the mutations from multiple selection experiments and ask if we can find, say, fitter phages that can kill these pathogens? And so here is sort of a simple growth curve of a urinary tract infection E. coli. So when you apply no phage, it grows just fine. when you apply the wild type T7 phage, there's an initial loss, but then a resistant strain emerges, and you see this here. But now when we apply our combinatorial mutant library of phages, it is completely dead. And so this looks promising. Now, it doesn't work in every UTI strain, but it's started to work in a subset of strains. And I'll mention one more example before I close out. And so this is this is against the highly multi-drug resistant bacteria. And so if you go to the CDC website, there is a group of bugs that they call as urgent threats. And one member of that group is this carbapenem resistant enterobacteria. And so some of you may have heard of this as the NDM1 superbug. And if you look at the ATCC spec sheet, it is resistant to like 35 antibiotics and only susceptible to 2 antibiotics, right? So it's up there among the deadly ones. And so again, we said let's see if our combinatorial mutant of phages is effective against this. And here's sort of a similar plot. Again, the bug by itself grows just fine. When you apply wild type T7 phage, it drops, but then it eventually sort of picks up, and then a resistant strain emerges. When we apply our T7 mutant, a resistant strain emerges, but because we apply a complex combinatorial mutant, we think that results in other mutants of the phage possibly blocking off a resistant mutant and ultimately killing the host. And so the takeaway from this is that we think we now have a rapid pipeline for generating these synthetic phages. And these synthetic phages, in some instances, do outperform the wild type phage many times over, and show promising activity against these multi-drug resistant strains. And so the stage is now really set to target other pathogens. And so we're pursuing a few broad directions in my lab. We're using all the sequence function data sets to learn to use machine learning to figure out what are the basic molecular rules for engineering phages. We're targeting different pathogen groups. I mentioned E. coli. We're targeting 3 or 4 other pathogens. We're super interested in using phages as tools to carry out precision microbiome editing. And so the goal here is to precisely remove offending members of a complex community. And so I've only mentioned mutations on the phage side. What we're also doing are sort of high-throughput experiments on the host side to ask where the Achilles heel of the phage lies. So I think I'm just going to stop there, and I'll sort of quickly acknowledge the people in my lab, who and all the funding agencies who supported our work. And I want to sort of give us 10 minutes to answer questions.
Thank you for the super interesting talk. That's some really cool stuff. So if anyone wants to ask a question, they can either unmute themselves and ask it, or they can send it to me in the chat.
One question. I was wondering if there might be T7 phage proteins other than the receptor that have evolved to be less than optimal for infectivity and killing of bacteria.
Yeah, there's probably plenty of place. The optimization likely has to happen on multiple genes, right? The polymerases could be slow. The promoter sequences may not be totally optimized. Yeah, so the receptor binding domain seemed like a good place to start, right? Because it drives so much of the phage activity. But you're absolutely right. There are plenty of other places.
And actually, I had a question too.
Yeah.
So I noticed that you're mainly looking at like single mutation differences. Is it possible? Is there any way you can use the Oracle system to look at making one change and then making changes based off of that? Or would you then need to like create a mutant and then apply the Oracle system to that one specifically? Or could you like explore the possible mutational space through sequential mutations in any way? Because I imagine that might make a difference in how they evolve.
Yeah, you're absolutely right. So we're doing both things. So since we have a ton of sequence information, there are obvious combinations that we can make and test, and I've shown you data on that. But we are also making continuous evolution systems where we can, phage is like the perfect continuous evolution system, right? And so so we're doing that too. So the goal there is to let it explore different paths. And the idea is, so we can sort of, to tie your question to the previous question, what we're hoping to be able to do is to continuously evolve several genes at the same time, right? And so the receptor binding domain would be one, but you can easily imagine 3, 4 other genes that could be evolved at the same time.
Okay, cool. Yeah. And then I think Professor Moon had a question too.
Actually, I wanted to wait until all students asking question, and then I will ask the question if we have time.
Okay, sure. So did anyone else have any questions before we get to Professor Moon's questions?
I guess I had one more. What are your thoughts on the efficacy of computational methods out there in predicting promising sites to mutagenize so as to like reduce the library complexity or library size? And like, I guess, would you have any recommendations maybe?
So I guess I'd say there are sort of maybe two broad classes of computational methods. One is sort of purely structural biology based methods, where Rosetta and other such approaches. Those could be partially effective in choosing mutations that are not destabilizing, right? So for instance, if there are mutations that are obviously destabilizing and we're dumb enough to put those in, I mean, that's you're wasting library space, right? So that's sort of, it could be helpful that way. The other class of computational approaches are sequence-based approaches. And so these are emerging in the last 2 or 3 years. And so the idea there is that you can look at the universe of all protein sequences and ask and look for common patterns, and then ask if there are interesting changes to those common patterns that could result in enhanced fitness. And people have started using that information for protein engineering. And so that would be really valuable. So, and I'll mention what we're also doing is we're using such approaches to specifically scan phage genome databases to ask what are the sequences that natural phages often use that we could sort of copy paste in here, right? Natural phages have done amazing selection, right? So that information exists. We just have to have the computational tools to harvest that information and put those back into this. And so just to sort of create the dichotomy, the structure-based approaches, sequence-based approaches, and they serve different purposes and would no doubt be beneficial.
Okay, thanks.
Any other questions?
So I noticed there's a question in the chat from Lai. And Lai is asking, do you think that we should incorporate unnatural amino acids to increase the number of protein mutants to further improve the current phage?
I think the short answer is yes. The longer answer is there is a lot of space to be explored with the natural amino acids that we've barely scratched, right? So unless one can make a compelling case for incorporating non-natural amino acids, I'd say it is a little lower on priority, right? That's the way I would think about it. So I mean, really, any increase in the search space, there's a trade-off, right? Because as you increase search space, then you're also searching junk sequences, right? So you have to be a little careful with how far out you go.
Okay, thanks. Did anyone else have any questions? Or maybe Professor Moon can ask his question now?
So,
okay, Professor Moon.
My turn? Okay, so amazing talk. I love it. So, and I also saw, you now expanded your research portfolio into interesting and important problems, including phages. And although your talk, today's talk is more on phage, I want to ask regarding protein engineering because you are basically a real expert in that field. Yeah. So my question is, what is your perspective on future or remaining challenges in protein engineering? To me, also, I believe the community think protein engineering is old technique or field, but I believe this is still very, very challenging problem. Even just changing, one, sensor, protein-based sensor, from one specificity to the other, or promiscuous enzyme or promiscuous transcription factor, one from the other, based on my own experience. And that's still a huge problem, but still now in the community kind of think this is an old problem, not that much interesting, and that's kind of ridiculous to me. Well, I just want to hear your perspective because this is still very challenging problem.
Yeah, so that's a great question, and I'd say there are problems that we've solved. There are bigger problems that we have not solved, I think you hit upon some key points here. The computational methods, what they've done over the last, I'd say, 2 decades now, is we have become really good at figuring out the rules that make protein structure, right? If you want, and David does this, and other people, David Baker does this, and other people do this. If you want the protein to fold into a certain shape, it's almost it's a geometrical problem now, right? So you can specify it and it does it, which is remarkable, right? Which is because this is, if you look at even in the late 90s, early 2000s, it was a pipe thing, but now you can draw a protein on the back of an envelope, and then you can synthesize a sequence that would make it and then folds it, so which is remarkable progress. But what we have not made much progress on is design of function, right? And catalysis and transcription factors, transporters. I mean, geez, that's so hard, right? And part of the reason for that is it's a protein is not simply a block, right? I mean, it moves. There is dynamics. There is allostery. There is epistasis. There is compensation. It's a very dynamic entity, right? And so, to what extent one can codify that in a computer program, it is not totally obvious, right? Now, the solution to that would be, in my opinion, would be sort of, I call what we do is as phage biology for dummies, right? Because I have no idea how it works, so I'm just going to throw the kitchen sink at the problem and see what nature tells me, and see if we can use tools to figure out the patterns, right? And so if one cannot solve the problem from first principles, then one can do pattern recognition, right? And so to me, that is a promising way forward, because you can take a catalyst or a biosensor or a transporter or something else and do these kinds of large sequence function screens. And if you do them enough times, you can, I skipped those slides at the end. If you do them enough times under different conditions, different substrates, then one can start piecing together the rules, right? So we can be smart about how we do design them. And so to me, that is the way forward, at least in the next 5 to 10 years, of how design of function can happen, right? So deep learning methods.
Yeah, that's absolute point. I wish you are, the journal editors, I mean, Nature or Science or Cell, because I mean, they now kind of think, this field is old, and then without, real computational component of the, protein engineering, they are not interested in anymore. So I have to, actually I have some problem when I submit my paper to their journal, but I'm okay with the current one I published recently, but that's kind of struggle. I mean, people now away from the protein engineering without any computational component, but we have the tons of protein and enzyme to evolve, and that requires lots of protein engineering. Still very challenging problem.
I agree.
Yeah, thank you so much.
Yeah, of course. Any other questions? Alrighty then. Well, thank you for the invitation.
Okay. Yeah. Thank you so much for speaking. That was an amazing talk.